drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a Ph.D. in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Start off by just telling everyone a little bit about who you are and what you study.
Yeah, my name is Nadal, as you said. Well, I really began to study chemistry in my hometown, which is Alicante in the southeast of Spain. And later I moved to the Autonomous University of Madrid to study biochemistry and molecular biology. And after that, I decided to come back to Alicante, where is an excellent institute of neuroscience, and I did my Ph.D. in that institute. Although I work with beta cells that are not really neuron, but from an electrophysiological point of view, we were interested in studying the stimulus secretion coupling in pancreatic beta cells and how the electrical activity of the beta cells drive to the calcium signaling that produces the release of insulin in those cells. So it makes sense to study that, being in an institute of neuroscience, because, in the sense that we were very interested in ion channels and calcium signaling and those stuff. So I did my PhD there, and later I moved to King's College London, UK, to do my postdoc there. So I stayed in King's College 4 years, and I was studying the connection between calcium signaling and cell division in astrocytes. So I was really in a group of neuroscientists and working particularly on that, how proteins such as albumin that were going through the blood-brain barrier, what the blood-brain barrier breaks down, activates astrocytes and make them to divide and produce a scar, and this sort of stuff. And really it was in London when I first learned about endocrine disruptors. That was at the mid-90s. And also about the extranuclear actions of estrogen receptors. So at the end of the 90s, I moved to Alicante because they opened a new university here, and they contact me to go back, let's say, home and start in this new university. And yeah, I decided to begin working in estrogens and endocrine disruptors in the pancreas, in the endocrine pancreas, the beta cells. So that's more or less my background.
Yeah, no, it's an interesting background, and even though we're going to talk probably mostly about the pancreas and endocrine aspects of the pancreas, hormone secretion, insulin, estrogens, things like this, and endocrine disruptors, I think it's worth reminding people that we'll probably talk about the brain to some extent at some point, maybe. But, even though we think about like the pancreas as an endocrine organ, and we don't think of it like a neural organ because it's obviously very different from the brain. At the same time, much of the brain, important parts of the brain that we might talk about are neuroendocrine organs themselves. They also secrete hormones. And, even though the pancreas isn't made out of neurons like the brain is, part of your studies were studying the electrical properties, ion channels, all these things that we normally associate with neurons are found in other cells as well.
Exactly. Well, I think I consider myself a beta signaler. And so the beta cell works very much as a neuron from that point of view,
Yeah.
that
Yeah.
it's an excitable cell, it produces action potential, and are these action potentials which code finally for the insulin release,
Mm.
very similar to what a neuron does uh, for neurotransmitter release.
Yeah. So yeah, let's talk about the pancreas a little bit just to give people some sense. You're referring to the beta cells. So these are a major cell class, if I understand correctly, they're a major cell class of the pancreas, and these are the ones that secrete insulin. You're actually saying that analogous to the way that a neuron will release neurotransmitters in response to a change
Mhm.
in electrical signaling within the cell, something similar is happening to regulate insulin release from these beta cells in the pancreas.
Mhm. Yes, the pancreas, we have two types of pancreas: the exocrine pancreas, which is not related uh, um, to the endocrine system, and the endocrine pancreas, which are islet of Langerhans are called, which is the basic physiological unit of the islet of the endocrine pancreas. And we have in humans million of them inside the pancreas.
Mhm.
So each of these islets contains between 1,500 to 3,000 cells of five different types. Uh, the majority of those five type of cells is the pancreatic beta cell. In human, it's about 50 to 60% of the islets are pancreatic beta cell, and about 40% are glucagon releasing alpha cells, and the
Mhm.
rest are minority somatostatin releasing cells and others. In mice, it's a little bit different. They contain more beta cells, so about 80% of the cells in the islet are beta cells, and they contain less alpha cells releasing glucagon, about 10%, 15%.
Mhm.
So that makes a bit of a change when we compare the situation in mice to the situation in humans.
I see. So one portion of the pancreas is devoted mostly to endocrine functions. So this is the one that
Yeah,
people probably hear about most that's associated with insulin and blood glucose regulation. Basically, you're saying that chunk of the pancreas is composed of what we call islets. So these are basically patches of tissue, each of which contains a certain combination of cells, such as the insulin secreting cells and others.
exactly. Yes.
And so I don't want to spend too much time on this because I've covered it on the podcast before. And I think people are most familiar with this part of the pancreas. And we're going to also talk about other things the pancreas does that I think people don't appreciate as much. But
Mhm.
can you give us just a very basic, very simple rundown of how these beta cells and cells in the endocrine portion of the pancreas regulate blood glucose levels via insulin?
Yes, the pancreatic beta cell is the cell that produces insulin. It expresses insulin and biosynthesizes insulin, and is the only cell really that is able to release this insulin in response to nutrients
Mm-hmm.
in our body. And that's very important because insulin is the only hormone able to decrease blood glucose.
The only one we
So
know about.
the only one we know about. So if the pancreatic beta cell goes wrong,
Yeah.
then we get diabetes.
Yep.
And that's why it's very important for all of us. Um, uh, so normally after a meal, what we have is an increase in blood glucose. And this is because the digestion and there is, of course, increase in glucose, amino acid, and fatty acids, and they go to the pancreatic beta cell. So the pancreatic beta cell, uh, if we focus in glucose, uptakes glucose, metabolize glucose through glycolysis and the Krebs cycle, and produces an increase in ATP ADP ratio. And this connects to the a channel at the plasma membrane, which is the ATP sensitive potassium channel that controls the resting membrane potential of the cell.
Hmm.
So when the ATP increases, this channel closes, potassium stops going out of the cell, and then the plasma membrane depolarizes, opens calcium channels, voltage gated calcium channels, and calcium gets in the cell and produces the exocytosis of insulin.
Hmm.
And then insulin is released into the blood.
Yeah, and this is like what you were talking about before. It really is a lot like neuronal transmission in the brain in many ways. There's ion channels that are coupled to ATP, ADP levels in the cell. So, metabolism is being coupled to channels that regulate the potential of the cell. And when the cell becomes sufficiently excited, certain ions come into this. So there's like a more abundant ATP. This causes things like potassium and calcium to flow back and forth between the cell. And at a sufficiently altered membrane, the cell will then release, in this case, insulin, not unlike the way that a cell in the brain might release neurotransmitters.
Yeah, exactly. That's it.
Interesting.
That's it.
Okay, so insulin gets released, and basically it's telling the body, take some of that glucose out of the bloodstream and store it in cells.
Exactly. Insulin has receptors in different tissues, but mainly it works at the liver, skeletal muscle, the adipocytes, and also in the brain.
Mm-hmm.
So what it's insulin says to the skeletal muscle, please uptake glucose, and indeed it's the skeletal muscle, the tissue that takes more glucose away from our blood, tells the adipocytes also to uptake glucose, and the excess of glucose is stored, it produces lipogenesis, and it tells the liver to stop producing glucose, which is the function of the liver in terms of glucose homeostasis. And it also tells the brain that insulin has been released and there is nutrients already there,
Yeah. So it's
not
serving
more food intake. Yeah.
Yeah. So the insulin's not only telling parts of the body, like the liver, to store the glucose and basically save it for later, but it's also telling the brain, like, basically that we just ate. It's important for instigating some of the satiety regulating mechanisms of the brain. It's saying we just ate something. There's nutrients. Prepare the organism to deal with that.
That's it.
And so this is a core mechanism of the body. It's super important that blood glucose levels don't get too high or too low. Insulin secreted from the pancreas. You're saying the pancreas is these are the only known cells in the body that secrete insulin. So it's extremely important in that sense. What is so important about preventing glucose levels from getting too high for too long? What are some of the things that start to go wrong if glucose remains elevated?
Yeah, if glucose remains elevated, it starts to affect mainly endothelial cells and the vascular system, and that begins to produce problems. And this is common. I'm not an MD, but I know a little about that, and it is common in diabetic people for many years to have complications with in the eye, for instance, the retina, and to have problems of vision, to have diabetic foot because of this lack of microvascular tissue, which is affected there. It affects the heart and makes you more have a higher probability of a heart attack and other things. So really, having high levels of glucose for a long period of time is nasty. So is to our organism.
Yeah. And on a sort of coarse-grained, intuitive level, I would imagine, glucose is fuel for the cell. It's like a primary fuel source for all of our cells. you don't want to give them too much too often. I guess, maybe an analogy would be like, I have my laptop here and it's plugged into the power source, but the power source has a specific voltage. It's sending a specific amount of power. If I was to hook this up to a massive generator that would send too much power all at once, I could blow out some of the circuits of my computer.
Yeah. Yeah, that's it. I mean, the point is we always need to regulate glucose very accurately, and the beta cell is prepared for that.
Mm-hmm.
So it's prepared to have a response in terms of insulin release to very small amount of insulin. It can distinguish between 6 millimolar to 6.1 millimolar,
I see. It's highly sensitive.
6.2 milli, very highly sensitive to those changes.
Mm-hmm.
And we need that to properly adjust the tissues that I just mentioned, like the adipocytes and the liver, etc., to control and maintain the glucose levels as constant as possible.
Mm-hmm.
Otherwise,
Yes, it's just a highly sensitive control system.
Yeah.
So I want to ask you, I know that I think this ties into a lot of the other stuff that we're going to get into, but basically a big part of the pancreas, as we said, is an endocrine organ. One of the major hormones that is being secreted is insulin. That's what we just talked about. But there's probably a bunch of other sensitivities here to other hormones and other signaling pathways tied to other hormone systems. Can you start to talk about some of the other hormones that the pancreas is sensitive to? For example, I know that you've done a lot of research on things like estrogens. So how do some of these other hormones fit into the picture when it comes to the pancreas?
Yeah, I mean, there are different hormones that can control the insulin release. Before entering into estrogens, perhaps it's easy to think about when we are stressed and adrenaline is released, that what it does is to decrease insulin response to glucose so that we have more glucose in blood to go running,
Yeah. If I need to run away
because
from something scary.
we need it,
Yeah.
Yeah, if we are in danger and we have to run. So adrenaline, of course, affects the pancreatic beta cells in that sense. And later there are other hormones that may have a more long-lasting effect, such as estrogens, for instance, which are the female hormones. And we are interested in how particularly estrogens affect the pancreatic beta cells because the islet of Langerhans, the endocrine pancreas, adapts to pregnancy. It has to adapt to pregnancy. This is a very interesting period, pregnancy, to study from the physiological point of view, but very difficult as well. But during pregnancy, a woman has an increase in tissue because of the fetus, the placenta, and all that is growing. And the pancreas, her pancreas needs to adapt to that increase in tissue. And so therefore, there's an increase in the number of pancreatic beta cells. There is also an increase in how cells senses glucose. They start sensing glucose to low levels and secreting more insulin. And in part, this is produced by estradiol and other hormones as well that are particularly going up during pregnancy as placental lactogens.
Interesting. So yeah, this makes perfect sense. So obviously, when a woman gets pregnant, there's a growing fetus inside of her womb. She's going to need to provide nutrients and energy to the growing baby, and so it makes perfect sense that something like this system would have to change because it has to adapt to that change in the woman. You're going to have to provide extra nutrition for the two bodies now instead of the one.
Yeah.
And if, to anticipate maybe some of the places we're going to go, it also makes perfect intuitive sense that something like an estrogen would regulate that process because estrogens are naturally going to be fluctuating over the course of pregnancy. And so all the stuff is hooked together so that there's these metabolic adaptations in response to these hormonal changes triggered by pregnancy. But I would also imagine if estrogens instigate those changes in the pancreas during pregnancy, changes in estrogens in other contexts would probably naturally alter pancreatic function as well. So if we're, for example, living in an environment that is highly estrogenic because we're exposed
Hmm.
to estrogens in the environment, that could potentially affect insulin signaling and pancreatic function in everyone, not just pregnant women.
Yeah, indeed, that was the idea when we started with our project using bisphenol A, which was known to be a xenoestrogen that was known to act as an estrogen,
Hmm.
as a weak estrogen, the people said. That was 25 years ago, something like that. So the idea is, okay, uh, if this is working like a estrogen is doing the same as the natural hormone to pancreatic beta cells in mice, so that we start doing is giving bisphenol A and estradiol, the natural hormone, separately to male mice.
Mm.
They never had to be exposed to these doses of an estrogenic compound, as you just mentioned. So, and then we discover something that was fascinating from the scientific point of view, but scary from the personal point of view too.
Hmm.
And is that bisphenol A at low doses was producing insulin resistance and hyperinsulinemia in mice.
Hmm.
And it was affecting insulin release and other peripheral tissues as well
Hmm.
in adult mice. That was the first
Yeah. So
study
basically,
we did. So,
basically, when
yeah.
we think about things like insulin resistance and type 2 diabetes, which we'll probably talk about in different ways,
Yeah.
normally, I think a lot of people, when they think about why someone becomes diabetic, they think about, if it's type 2 diabetes, at least, that this is induced by the diet, that it has to do with sugar and carbohydrate intake. And indeed, those things are very important. But I think what you're starting to say is that you can get insulin resistance by other means. You could be exposed to certain hormones, for example, or certain endocrine disruptors, and even in the absence of, say, drinking Coca-Cola every day, you can still develop
Mhm.
insulin resistance in those ways.
Yeah, exactly. I mean, what I think we know now is that many of these endocrine disruptors that we study are part of the diet as well.
Mm.
For instance, bisphenol A, phthalates, and other bisphenols are part of our plastics
Hmm.
components, and I mean they are in food contact material. So when we buy something that is involved in plastic, uh, so this plastic is going to release these bisphenols, phthalates, and other uh, chemicals. You
Hmm.
know, plastic contain hundreds or thousands of chemicals.
Hmm.
Indeed, it's not only a polymer.
Yeah.
And I think this is part of our diet as well. So we always think in okay, lipids, sugars, and of course, as you said, this is very important, and it's no doubt that they affect, insulin sensitivity, insulin release, and glucose homeostasis, and are obesogenic in general, the excess. But there are other chemicals in food coming from other sources,
Yeah, and
that we ingest.
for better or
Yeah.
worse, these things are in our food supply, and to some extent, they're unavoidable. A lot of this stuff is fascinating, but a lot of the stuff, the stuff you work on is fascinating, but it's also terrifying. So a lot of the stuff is going to be pretty unsettling, maybe to people who haven't looked into how widespread these endocrine disruptors are. But
Mhm.
before we really dive into some of that stuff, I want to talk a little bit about estrogen signaling and estrogen receptors and sensitivity in the body. So obviously, the body has natural estrogens that it produces. As you mentioned, we've got BPA and other xenoestrogens, artificial estrogens that we're going to talk about. Can you say a little bit about estrogen receptors? How many estrogen receptors are there? And is there anything sort of special or notable about them in terms of their sensitivity or how they work that's going to be important for
Mhm.
understanding some of the other things we're going to talk about?
Yeah, there are three estrogen receptors. Two of them uh, that are the more classical ones are estrogen receptor alpha and estrogen receptor beta.
Hmm.
They belong to the so-called family of nuclear receptors, and they classically act as transcription factors.
Hmm.
So they bind the ligand, the natural hormone, in this case 17-beta estradiol. They dimerize, they go to the nucleus or are already in the nucleus, depending on the cell, and bind to an estrogen response element in the DNA, and that puts all the machinery of transcription to work,
Hmm.
okay, and increases gene expression. And there is another type of response, which of these very same receptors, which is not in the nucleus. It's what we call extranuclear initiated effects. So in the last 30 years, there have been many different studies that have been published to show that these receptors, ER alpha and ER beta, are able to activate kinases outside the nucleus, such as PI3 kinase, ERK1/2, MAP kinases in general, and then to regulate other proteins or signaling pathways, including ion channels.
Hmm.
So we have like two ways the cell respond to estrogens in terms of estrogen receptors. The
I see.
nuclear one, which is the very classic one, and the extranuclear, which is rapidly initiated and activates as signaling. And this one is very particularly important in terms of endocrine disruptors.
I see. So oftentimes
And
when we
yeah,
think about a receptor, they're often located on the outside of the cell, and the cell responds to something on the outside, and this leads to changes
yeah,
on the inside of the cell. But for for these estrogen receptors, they are internal receptors. They're either located
exactly,
in the cytosol or in the nucleus. And in the
exactly.
nucleus, when like something like an estrogen binds these nuclear receptors, that's directly leading to transcriptional changes. So genes get turned on or turned off, things like that.
Yeah, exactly. And later, there is one more estrogen receptor that was proposed about 2005. Uh, and it's a G protein coupled receptor. It's called GPER.
Mm-hmm.
It's at the membrane. And it seems that it binds estradiol as well and have produce estrogenic signaling.
I see. So
This
you've
one
got,
is less studied, but
yeah,
it seems that it's also there at the membrane. So we
yeah,
have
three
the two
layers of
nuclear
control.
receptors, exactly, and one at the membrane. And they crosstalk, so that makes life complicated.
Yeah,
But
yeah. So, estrogen is flowing through the bloodstream. It can get into cells. It can get all the way through the membrane into the cell, through another membrane, that's the nuclear membrane, and it's going to
mhm.
regulate gene transcription in the nucleus. It can also get through the outer membrane, but not pass through the nuclear membrane and regulate what certain proteins are doing in the cytoplasm. And there's also potentially these estrogen receptors on the outer membrane. And so you've got like three interacting levels of control inside individual cells
Yeah, to the same hormone, to the
for the
same
same
signal.
hormone. Yeah.
Yeah.
And is that special about estrogen, or is that sort of a normal thing when it comes to sex hormones generally?
Uh, it it's now known that it's not only for estrogens. I would say for steroid hormones, we can say that, and there are the nuclear receptors act outside the nucleus as well for androgens, progesterone, vitamin D as well, and whether there is G protein coupled receptors, that's a different story.
Mm-hmm.
Has been proposed for some, uh, but not for other steroids. But
Yeah.
it is very well accepted, I would say, that nuclear receptors act as transcription factors in the nucleus and also outside the nucleus uh, for most steroids
Mm-hmm.
hormone.
And are most are most tissues in the body sensitive to estrogen to some extent, or is it highly concentrated in a specific subset of tissues?
I would say that all tissues in our body are sensitive to estrogens.
Yeah, yeah. And
Yeah,
I mean, I
yeah,
guess that makes
in
sense.
general.
Yeah,
Yeah,
I guess that makes sense because, if you think about something like just the sex hormones in general, they are inside of everyone. They fluctuate across the lifespan. If you think
mhm.
about something like puberty, right, the whole point of puberty is you need to transform all of the tissues in the body in a certain way. And so I guess these hormones are important for initiating that transformation. And it makes sense that, more or less every tissue is going to be sensitive to some extent.
Yeah, yeah. But I think it makes sense, and perhaps there is some tissue that it doesn't have some estrogen receptor or estrogen receptors, but I'm not aware of it.
Mm-hmm, mm-hmm.
It is true that this we have these three receptors, the two nuclear and the GPCR, and it is true that each tissue may have different level of expression of
Yeah,
each
yeah.
receptor. That is true. But as far as I know, most tissues express estrogen receptors.
Mm-hmm. And I want to start to talk about endocrine disruption a little bit. I don't know if there's anything you want to set the stage with, but I suppose an endocrine disruptor would just be any exogenous molecule that can affect hormone signaling in an unnatural way. Is that a fair basic definition?
Yes, this I would say is the definition of the endocrine society. Indeed, is uh, an exogenous chemical, which can be natural or uh, or not,
Mm-hmm.
or artificial, but it affects hormone action.
Mm-hmm.
That's the more extensive definition of endocrine disruptor.
Yeah. And so how did you get into this field, and why has this become such an important area of study?
Yeah, when I started, really was at the end of the 90s when I came to Spain back from England, from London after my postdoc, and I had to start my own research line. So I was starting. Really, my question was how estradiol has a role in adapting the pancreatic beta cell to pregnancy.
Mm-hmm.
So then I was at home with my wife, and we were watching TV, and we watched a TV documentary that was named Assault on the Maid. It was a BBC Horizon program,
Hmm.
and one of these documentals. And most of the pioneers in the field of endocrine disruptors appeared there.
Hmm.
And they were explaining, how these endocrine or these chemicals were acting through nuclear receptors to affect different tissues. Of course, nothing about uh, obesity or diabetes at that
Mm-hmm.
time. And because we were using in the lab estradiol and see the adaptation of beta cell using estradiol, say why we don't start measuring what happened with this compound called BPA, that
Hmm.
is phenol A, that at that time was not so famous as it is now. That was in 97, 98. So I tested measuring ion channels, measuring the KTP uh, channel activity with patch clamp technique. So I acutely added bisphenol A at a very low concentration, 1 nanomolar, and 5 minutes after uh, perfusing the cells with bisphenol A, the channel closes.
Hmm.
Uh,
So a tiny
and
amount
I
of
said,
BPA had a big impact.
tiny amount. This is the amount we have in blood.
Oh, this is that.
And I
This is the
said,
amount
well,
that's in our bloodstream
yeah,
potentially right
yeah,
now.
it is between 1 and 18 nanomolar BPA.
Wow.
And that has been measured and is more or less the amount of BPA we may have in blood.
Hmm.
And then we started. We that That's
Yeah.
how
So
we.
you saw this documentary and that made you want to do an experiment with BPA, and then you got started down this whole path.
Yeah, yeah, that was it. So yeah,
What let's explain for people very explicitly what is BPA and where does it come from most commonly?
yeah. Bisphenol A is one of the main is the main component of polycarbonate plastic.
Hmm.
So polycarbonate plastic, which is normally the transparent plastic that we have is hard. For instance, these glasses, this is no glass anymore, is polycarbonate.
Hmm.
So this is made by bisphenol A, and this is the polymer. And it's in many epoxy resins. It's in the interior of canned food. I mean, there is a plastic in there
There's a
in
plastic
contact.
liner in
Uh,
metal
uh, uh,
canned foods. So most people probably don't even realize that.
exactly. Yeah, it is made. It contains bisphenol A, and bisphenol A is used also as a plasticizer that makes the plastics softer, so you can give a shape to the plastic, etc. Otherwise,
Hmm.
the some of the polymers are very hard, and it's difficult to give a shape, and you need to solve the plastic. So BPA is added for that.
Hmm.
So BPA is practically in any plastic in a way or another. It's in PVC,
It's
yeah,
all over.
all over, all over. That's one of the problems. These, indeed, I want to be, these are recent results from a big European consortium uh, that is called Human Biomonitoring for EU. Um, they released all the data in the last year. You know, they have been studying the levels of chemicals in the whole European population. And the conclusion is that all Europeans above 18 have BPA in their blood
Hmm.
uh, at low doses, at least.
Yeah. But you said
That means
earlier that,
about 1 nanomolar, you
yeah.
know.
So is it safe to assume that virtually everyone listening to this has some amount of BPA in their bloodstream, and it's even if it's a quote unquote small amount, it's probably a physiologically active amount?
I would say so. Unfortunately, I would say that more than 90% of the people, uh, they at least in the US
Hmm.
and in Europe, where it has been measured, um, yeah, have BPA in blood, probably at a level that affects cells.
Yeah. So this isn't necessarily a thing where BPA is bad, but you need a very high dose of it, and trace amounts, are probably safe. This is a thing where potentially even low amounts could be problematic or could be having some physiological impact.
Exactly. That we see is that low amounts affect cells,
Yeah.
in
Yeah.
particular insulin releasing cells. So
Yeah. And I want to dig into that a little bit. So, from your studies and from what's known in the field, what is BPA? What kind of effects is BPA having with respect to insulin signaling?
yeah, with BPA, what it does is to first we can divide in two different uh, set of times. Let's say acute effect. So if we apply BPA, we very rapidly increase insulin release
Hmm.
in a matter of minutes. And this has been studied in isolated beta cells from mice, from human as well. And we know that it's mediated through the closure of the KATP channel. And it has been also demonstrated in humans by drinking just one dose of BPA, that it affects um, insulin release.
Yeah. So within
And
minutes, it's already doing something.
within minutes, it's already doing something. Yeah. Uh,
Hmm.
and later, we have done experiments more long lasting, let's say in vitro, in culture cells during 48 hours, exposure to 1 nanomolar BPA. So they regulate genes, they regulate insulin gene expression, for instance, and they change the content of the insulin, the pancreatic beta cells, increases the content of beta cells. They regulate potassium channels, calcium channels, and sodium channel expression at the membrane, and also the function of those channels. And and this is associated with an increase in insulin release.
Hmm.
So what BPA does is to produce an over-secretion of insulin.
I see. So it's not only immediately affecting the release of insulin, it's also having longer lasting changes that increase the beta cell's capacity to release insulin.
Exactly. And this is not only in experiments in vitro that are very good for understanding the mechanistic insights, but also in mice. I mean, if we inject with BPA or we give oral BPA to mice, they get hyperinsulinemia, they get insulin resistance, glucose intolerance, uh, high levels of free fatty acids, high levels of leptin as well, etc. in adults, males.
Mm-hmm.
Uh, yeah, and then there are other studies done during pregnancy, so we can talk later about that as well.
Yeah, but I guess
You
on a
wish.
basic level here, BPA is having a sort of pro-insulin effect. It's causing more insulin release. It's doing that
Yes.
immediately, and it's also leading to long-lasting cellular changes, such that the cells are basically able to release more insulin more often. And this is obviously going to tend to make it easier to develop things like hyperinsulinemia and insulin resistance. And if I can anticipate where this might go, um, this would suggest that let's say my great great grandparents,
Mm.
they were fairly metabolically healthy. They didn't have insulin resistance and things like that. Even if I was eating the same, someone today could be eating the same diet, the same nutrients in terms of fat, carbs, and proteins as, say, their great great grandparents. But if their great great grandparents were not exposed to BPAs and other endocrine disruptors, but you were living today, you might develop something like insulin resistance and diabetes in response to the exact same diet because your body is now more predisposed to do it.
Yes, I would say that's correct. Indeed. I mean, we have some chemicals. BPA is one example. There are others uh, that affect insulin sensitivity in peripheral tissue, including the skeletal muscle, the liver, and the adipocytes,
Mm-hmm.
and affect insulin content and insulin release.
Mm-hmm.
So are affecting your glucose homeostasis, and then in response to even the same diet as your grandparents, you may have a different control of glucose in your blood.
Yeah.
That's true. So
Yeah. Wow. How so when things like type 2 diabetes and insulin resistance are starting to develop, and even when they've developed, considerably, how easy is it for the body to reverse that? If you remove things like BPAs, if you change the diet, like the glucose and the carbohydrate intake, are these states, hyperinsulinemia, insulin resistance, things like this, are they reversible?
yes, it depends on the time. I mean, it's a very complex issue. Perhaps we have simplified a little bit, just to make it understandable,
Mm-hmm.
to be able to understand. But yeah, I think the point is for how long you are having those changes,
Yeah.
and depending on the time that you have the decrease in insulin sensitivity or the pancreatic beta cells overworking or whatever, it depends of how long that it can be reversible or not. So, for instance, we have done with adult mice, if we inject bisphenol A during 4 to 7 days, so a week,
Mm-hmm.
we get the metabolic changes. And later, we just don't inject for 4 or 5 days, and everything is reversible
I see.
in adults.
In adults.
in adults.
So at least at the early stages, it's fairly readily reversible if it's
Yeah,
just starting.
yeah. And this is something that happens as well with, for instance, people that is overweight or obese,
Mm-hmm.
and they have a decrease in insulin sensitivity independently of BPA and and the hyperinsulinemia. If they lose weight, even less than 10% of weight loss improve the metabolic parameters very
I see.
much.
So
And
even something like a less than 10% weight loss can lead
yeah,
to better
so
insulin sensitivity.
yeah, and if you start to exercise and to take care of this situation a little bit, you can improve. Uh, so yeah, in the case of BPA in adults, it is reversible. But it's something different in the case that during pregnancy you get an exposure to endocrine disruptors such as BPA or others.
Mm,
During pregnancy, it will affect the fetus and the mothers. And then the effects are long lasting for
I
the
see.
fetus and for the mothers as well,
So
for
basically,
dams.
yeah, if a pregnant woman is exposed to certain endocrine disruptors at certain phases of development, I would imagine there's certain sensitive periods. This can basically lead to permanent changes that will be lifelong lasting for the baby that's born.
Exactly, exactly. So that's, I mean, uh, the fetal development is when most genes are, uh, expressing and stopping expression and
Yeah,
all
yeah.
this kind of stuff. And then is the most susceptible period for this kind of endocrine disruptors acting through nuclear receptors, you know,
Mm-hmm.
uh, to make epigenetic changes and then altering the
Mm-hmm.
uh, you, you know uh, uh, gene expression later in
Yeah.
life as well, altering the phenotype later in life.
Does BPA or other endocrine disruptors, do they actually cross the placenta and get directly into the fetal bloodstream?
Yes, indeed, BPA does, and others such as dioxins they do as well. And most of the lipophilic uh, endocrine disruptors are able to do that. So they
Mm-hmm.
pass the placental barrier and go to the fetus and signal there.
Mm-hmm. And I think you said that. So not only can it lead to um, permanent, long-lasting changes for the developing fetus that will go all the way into adulthood. I think
Mm.
you said also that it can lead to long-lasting and permanent changes for the mother. So the mother's own endocrine system can be disrupted such that it sort of doesn't go back to normal or back to baseline after the pregnancy ends.
Yes, I mean, this is less known that the effects on the fetus, but for instance, during pregnancy, if mice are exposed to endocrine disruptors, so we did it first with BPA, we already published that in 2010, and later have been done with dioxin as well. Um, then you get a change in the hormones in the mother
Mm-hmm.
during pregnancy. So it's not only passing the placental barrier and going to the fetus, it's that it's producing an, a change in insulin levels in plasma, leptin levels in plasma, and lipids in the mother. And that changes the environment for the fetus.
Yeah.
So it's not only the endocrine disruptors passing, but the whole environment in the mother is changing. And later, if you study those mothers as we did, everything goes back to normal during 3, 4 months. And after that, the mothers started to be overweight, insulin resistance, glucose intolerance. They have decreasing beta cell mass, decreasing insulin release from beta cells, and all kind of metabolic alterations in dams. You know,
Yeah. Wow.
and this is pretty new. It needs to be very much studied, but I think it's like a new uh, sensitive window for the mother.
For the mother.
I mean, yeah, pregnancy is a very
Yeah.
plastic period for the
Yeah.
mother as
Yeah.
well. And I mean, we have a project now uh, that we're going to try to understand what mechanisms are taking place in the mother to have these long-lasting effects that
Yeah. Yeah.
appear later. Even they disappear and they appear for months afterwards.
Yeah, yeah. And immediately you wonder, it's difficult not to wonder about common experiences that certain women have with pregnancy. So, for example, some women have a child, one or two or three children, say, and they have no trouble or very little trouble losing weight and getting back to where they were before the pregnancy. And other
Mhm.
women struggle with weight gain that doesn't go away and things like this. And one wonders if this ties into that, if it has something to do with exposure to things like endocrine disruptors like BPA.
Yeah, of course, this is pure speculation, but I mean, what I can say, our experiments is that mice that were exposed to BPA during pregnancy, uh, later in life, they were overweight and had these metabolic problems. And we did a control, just exposing for the same time non-pregnant females.
Yeah.
We exposed to BPA exactly the same time, and they had no overweight and no metabolic problems.
Yeah.
This is what I can say. I don't know if that, I mean, we need to
Yeah. We don't know if
do
that's true in
studies,
humans for sure, but
exactly,
yeah.
but it's a possibility, at
Yeah.
least that
But basically what you're
we
saying
need to
is,
think about.
yeah. So pregnancy is basically, and we're talking about mice here, but by analogy, it makes sense that this would apply to other mammals. Pregnancy is sort of like it's a critical period in the adult mother. Normally, we think about critical periods of development. We think about childhood and infancy and things like this. But in the
Mhm.
adult mother, things are basically more plastic while she's pregnant because obviously her whole body needed to be plastic, needed to rearrange itself in
Mhm.
order to accommodate the fetus. And so this can open up the opportunity for long lasting changes to happen in a pregnant female animal that would not otherwise happen to a non pregnant adult female animal.
Yeah, exactly. That's a very good summary indeed of what
Wow.
I try to say. Yeah.
It's yeah, I mean it's just it's fascinating. It's simultaneously fascinating, but obviously very unsettling.
Yeah, it is scary. I mean, it's always you have this mix of feeling, when I see
Yeah,
those
yeah.
results. That's true.
And I want to ask you about sort of the, you've already said that BPA is pretty potent. Can you say a little bit more about like the dose and the potency of BPAs and other xenoestrogens compared to natural estrogens? Are these things generally more potent than natural estrogens, less potent? How do you start to think about that
Hmm.
stuff?
Yeah, I think that's a very important question actually, and one of the main problems that I think we need to still to understand.
Mm-hmm.
Let's say, for instance, bisphenol A and other bisphenols, they have a low affinity for estrogen receptors,
Mm-hmm.
about 500 to 1,000 fold less than estradiol.
Got it.
Okay, but uh, they act through these receptors at doses at the same level as estradiol. So let's say the affinity for bisphenol A of estrogen receptor is within the micromolar range.
Yep.
But we have effects, we and others, we have effects at 100 picomolar, 1 nanomolar,
I see.
which is far below the affinity of the receptor for
Uh, so it's
the
acting
ligand,
by it's acting
for
through
the
other
BPA.
means.
Well, I mean, we are working on that, and yeah, we are trying
Mm-hmm.
to publish a paper in a good journal now. And
Mm-hmm.
that we found is that mainly bisphenol A is acting through estrogen receptors outside the nucleus. Nuclear
Mm,
receptor, but through the extranuclear initiated pathway.
I see.
And when they do that, they have a very high efficacy.
Ah,
because there are ways to amplify the signal. That's one thing.
Hmm.
And the other is that there is a, a proton transfer from the ligand. This is what we're trying to publish, is some new results now to activate, to switch this extranuclear pathway.
Hmm.
So it's like having a kind of different mechanism to activate this extranuclear pathway compared to the nuclear pathway.
I see.
So the
So
nuclear pathway is uh, normally has to work with a high affinity
yeah,
ligand,
yeah,
and outside the nucleus, it can work with a low affinity ligand,
I see.
being,
So the
being with a high efficacy.
Yeah. So the basic idea here, if I'm hearing you correctly, is so we said before you've got these three levels of control in terms of the estrogen receptor. You've got intranuclear receptors in the nucleus, you've got
Mm-hmm.
receptors in the cytoplasm on the inside of the outer cell membrane, and then you've got
Mm-hmm.
receptors on the outer cell membrane. Even though these BPAs might have a lower affinity, a lower stickiness for the receptors than natural estrogens, because you've got these three levels of control, these three different types, these three different flavors of estrogen receptors, the BPAs might be acting very, very potently at that outer layer so that they can have strong effects even at a low dose, even though their affinity for the receptor is actually less than the natural estrogens.
Yeah, exactly. It is easier. Well, it is easier if the receptor is outside the nucleus,
Yeah,
interacting with other proteins such as kinases, and then produces second messengers. The signal gets amplified,
amplified. Yeah.
and this is something that it doesn't happen in the nucleus,
Hmm.
uh,
I
because
see.
yeah, there of course is one ligand, one receptor, and one estrogen response element in the DNA. So
Yeah.
there is no amplification in that sense. But outside the nucleus, you can interact and produce, for instance, cyclic AMP and produce many cyclic AMP molecules with just one ligand, and then have many uh, pKa
Yeah. Yeah.
activation and
I see.
phosphorylation, etc. So the signal gets amplified. And in
Yeah.
addition to that, so we think that there is another switch to activate these extranuclear effects, at least with BPA, that is more kind of a chemical reaction that's only the shape of the molecule inside.
I see. Yeah. So it's like at these different layers of cellular regulation, the signal can get amplified to different extents. It's sort of like
Yep.
so you said the nuclear receptors. It's more like a one-to-one thing. It's like you and I talking here. I could ask you to do me a
Yeah.
favor, and I'm just asking on Hal to do me a favor. It's one
Yeah.
person. But if I go to my balcony and I shout to a group of 10 people, I can now amplify my signal tenfold. And that's sort of like the outer layer of signaling here.
That's a perfect example. Thank you. Yeah.
Okay, but the point is basically, because there's this sort of question that I think some people have that I've had when I think about some of this stuff. It's like, okay, we've got all these estrogen receptor, these estrogen, exogenous estrogens in the environment. The environment seems to be net estrogenic. It's, when we look at sort of coarse grained signals in men and women, both of them seem to be drifting in the estrogenic direction. But the question in my mind was, well, if we've got these xenoestrogens, but they're actually less sensitive, they're less potent, or they have less affinity for the estrogen receptor than natural estrogens, they might have a net anti-estrogenic effect if they're sort of pushing the natural estrogens out of the way and they're less potent. But basically, what you're saying is that, at least in the case of BPA, because of signal amplification and things like this, they can have a net estrogenic effect, and that makes sense.
Yeah, it may have an estrogenic effect, but it not always does exactly the same as the natural hormone.
Mm.
That's another point.
Yeah, it's not having the
Uh,
exact same set of
no,
effects
no,
as the natural
not at
hormone.
all. No,
Yeah,
no, no, indeed, because well, the receptors they need to dimerize to produce, for instance, and you have homodimers of estrogen receptor alpha, homodimers of estrogen receptor beta, and heterodimers estrogen receptor alpha and beta. Okay, and the ligands can make a different pool of these dimers. So we have studied that, and it seems that bisphenol A doesn't produce heterodimers, but estradiol produces heterodimers of ER alpha and ER beta. So because of the pools of dimers is different between estradiol and BPA, that may be a reason to have different signaling. So some responses are going to be equal
Yep.
between both ligands, and some are going to be different.
I see. So that, I think that's
Because it's not
yeah.
exactly the same, and that's
Yeah.
an important point.
Yeah, that is important. So the exposure to these xenoestrogens, it's not a quantitative change in estrogen signaling. It's not just sort of boosting up or down
Mhm.
natural estrogen signaling, period. It's a different pattern of signaling and stuff happening in the cell that's
Yeah,
not just a different amount of estrogen signaling.
yeah, exactly. This is I how I look it personally is okay. Really, what BPA and other xenoestrogens is doing is altering, disrupting the estrogen signaling
Yeah.
somehow. So it's not really mimicking or antagonizing completely, but is
Yeah, it's truly a disruption.
it's a disruption. It's just making a different signal and then modifying the signal for the natural hormone because
Yeah.
BPA is messing around.
Yeah. Um, so obviously, when we think about it makes sense that we're talking about estrogen signaling in the context of things like pregnancy and early development. But of course, all of these hormones, estrogens, androgens, everything's in males and females, just, it's in different amounts. There's different patterns of fluctuation at different periods of the life cycle. We talked about pregnancy and fetal development a little bit, but I want to think about how these things can affect males versus females now. And it's natural to imagine that the effects would be quite different because the pattern of estrogen receptors and other things is going to be distinct in males versus females. What do we know about effects in males versus females for these estrogens like BPA?
Hmm. Uh, there is sex differences. This is true. I would say for BPA and yeah, the experiment that have been done in the past uh, in terms of glucose homeostasis or obesity, uh, I think the effect, at least with BPA, is more in males than in females. It seems that females are protected, but the studies were not really designed to study different in sex. But new studies have been done, for instance, with dioxins, and it is not a xenoestrogen, but it's acting through the aryl hydrocarbon receptor, which is another nuclear receptor. And these are results from Jennifer Bruin laboratory in Carleton University in Canada. And they show very well how the phenotype in terms of glucose homeostasis is different between uh, males and females. And they designed the study
Mm-hmm.
to follow males and females. And it seems that in the case of dioxins, uh, the action is mostly in females. They have a more dramatic phenotype that males
Yeah.
actually.
And I guess
So yeah,
I
yeah,
guess on a very basic fundamental level, we would expect there to be a lot of sex differences when it comes to endocrine disruptors because the
mhm.
differences between the difference between a male and female comes from it has an endocrine origin, right? It's the
Yeah,
endocrine system is
yeah,
actually
yeah,
sculpting the bodies differently and making them
yeah,
differentially sensitive to all of these
yeah,
hormones. So we should expect that, I think, right?
yeah. Indeed. I mean, for instance, in our experiments, we have never seen that phenotype of insulin resistance, glucose intolerance, or hyperinsulinemia in females.
Hmm.
So this is why we
In non-pregnant
started to do
females.
it in non-pregnant females, of course. I mean, in offsprings, I
I
mean,
see.
or in adults
I see.
when we treat
So,
it.
so you've got results where
Um,
BPA exposure in adult males and adult
Males.
non-pregnant females,
Yeah,
you get bad effects in the males that you don't see in the females so
we
much.
don't see in the females. We didn't publish in the females. We just didn't see anything and just mention very phrase, at that time. That was
Wow.
years ago. But other people have done later, these experiments. And it seems that in most of the cases, in offsprings, uh, the phenotype is in males and not in females. So
Yeah.
that has been done with BPA by the group of Rebecca Simons in the
Yeah.
Penn University there
Yeah.
in the States recently. Yeah, and in vitro cells, we have more or less the same. If we measure, for instance, apoptosis in the beta cells, we get with bisphenol A, but we don't get apoptosis with estradiol.
Hmm.
And indeed, estradiol protects from apoptosis by BPA produces apoptosis.
Interesting.
And they both use estrogen receptors to do this effect. So,
Wow. So obviously, yeah, you get different effects in males and females. Not too surprising. When we look at, stepping back for a minute, when we look at our modern environment, it seems to clearly be net estrogenic for males and females. I know that at least in the U.S., the age of puberty keeps getting earlier and earlier for females, and obviously
mhm.
in males, it's quite clear. Testosterone levels keep going down, fertility levels keep going down, sperm counts, et cetera, et cetera. Why is it? So we hear a lot these days about endocrine disruptors and xenoestrogens. We hear a lot about exogenous estrogen based endocrine disruptors. I don't hear a lot about xenoandrogens. Is there something? Why is it that there seem
Mm.
to be so many exogenous estrogens but not androgens? Is that actually true, or is there a reason for that?
I, I think the reason is that mostly estrogen has been studied.
Okay,
And for instance, the effect of some of these chemicals on androgen receptor is like opposite, is like antagonizing
I
uh,
see.
the androgenic effect. For instance, phthalates is a classical example since the 90s that they act as anti-androgenic uh, uh, chemicals.
I see.
And so they, let's say, decrease the response to androgens. Uh, so that's the point. As far as I know, yeah, I think estrogen receptors are the most studied, very likely because they were the first receptors
Yeah,
to see an effect, and
I see. But
you
for
know,
so basically the estrogen receptor and xenoestrogens, they might just be more studied, which is why we
yeah,
so a lot of
yeah,
this could be an artifact of where we're focusing our research energy. But the environment still does clearly seem to be net estrogenic, and at least what you're saying when it comes to phthalates is that, even though they're not estrogen disruptors like BPA, they have anti-androgenic effects.
yes, indeed, they do, and we know some of these chemicals they also affect uh, thyroid signaling,
Mm-hmm.
so they can perhaps not directly affecting the thyroid hormone receptors, but other enzymes that produce thyroid hormone.
Yeah,
And we know that there are chemicals that can also interact with other nuclear receptors, such as PPR gamma, where tributyltin is one of the main examples, I would say. It's an obesogene as well, and acts as an agonist of PPR gamma.
mm-hmm.
And yeah, there are dioxin, as I said, through the aryl hydrocarbon receptor, uh, etc. So
Yeah. So two of these other endocrine disruptors you've mentioned, dioxins and phthalates, where do those come from in the environment?
well, dioxin is a byproduct of industrial, when you burn paper or plastics or
Mm,
whatever, and you produce these dioxins. Uh, so it's an industrial byproduct, actually. And phthalates are in plastics.
it's another microplastic.
It's another microplastic, and I mean, it's another chemical in plastics. So phthalates are plasticizers, and there are also in many cosmetics.
So people are
Uh,
putting them onto their skin on purpose.
yeah, yeah. I mean, it's stabilizes perfumes, for instance.
Oh, so you could
Uh,
breathe it in. You could literally breathe it in.
yeah, yeah. That's how it is. Yeah, and yeah, there are really many of them of phthalates around, and the other one that is really widespread and is a problem are perfluorinated compounds,
Mm,
which are persistent. They're very difficult to metabolize. They're very difficult
I
to get
see.
rid of
So
them.
they don't like break
Uh,
down. They don't get excreted easily.
Not at all. So we don't know that there is not much about uh, whether they work through nuclear receptors or not. There are some papers say yes, others say no. But these perfluorinated compounds are a real problem, and they are also involved
Mm,
in altering our metabolism. So they
mhm.
produce obesity and diabetes as well
Yeah. Um,
in
I want
mice.
to I want to talk about obesity and energy intake and metabolic rate a little bit. So
Mhm,
when obviously we live inside of a massive obesity epidemic. Obesity rates have
mhm,
kept going up and up, and everyone has their favorite explanation for obesity.
mhm,
Most people think about it in dietary terms. Some people say we're just eating too many calories. Some people say no, 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 it's we're eating the wrong calories. It has to do with specific fats or sugars or what have you.
mhm,
But obviously, this whole field of study, the endocrine disruption angle, starts getting you thinking about ways that things like obesity and other metabolic syndromes can be induced independent of the food component. I shouldn't say food components, but the nutritive food components that we eat. You could disrupt metabolism with some of these chemicals in ways that could
mhm,
lead to something like obesity, even if you're eating the same diet as a lean person.
mhm.
What do we know about the effects of BPA or other endocrine disruptors on things like um, hunger levels, satiety, signals in the brain, gut hormones, all of the stuff that we normally think about when we think about food intake?
Mhm. Yeah, well, that's a difficult question. I think, yeah, obesity is the theory of obesity is a very difficult problem, and I would say it's not solved. Nobody really knows why obesity is rising at the way it is at the moment, and really how obesity is produced. Uh, I think what endocrine disruptors do is to increase our predisposition to be obese.
Mm-hmm.
So probably endocrine disruptors are not going to make you obese if you have a diet, uh, you know, with no excess of calories or not too much fructose or free fatty acids or whatever, and you exercise. So you probably are going to keep lean and metabolically healthy.
Mm-hmm.
That would depend, of course, of your genetic background as well. So there are multiple factors there.
Yeah.
So, but the point for me is that endocrine disruptors are one more factor in in the scenario there that make you more predisposed to be obese.
Yeah.
Uh, how that can happen? We need to imagine that we are normally exposed to a cocktail of endocrine disruptors, not only to one of them,
Right.
not only
Right.
to bisphenol A. If you are only exposed to bisphenol A and it's, for instance, affecting your food intake rate, probably you're going to adapt, and then you're going to have
Yeah.
less
It's
storage
easy
or
for the body to
whatever,
adapt to
easy for
one.
the body to adapt.
Yeah.
But we are exposed to about 40, 50 at least different chemicals that
Yeah.
have
Dozens and
been measured
dozens.
in our blood. And they affect every term in the energy balance equation,
Yeah.
which is energy storage rate equals energy intake rate minus energy output rate.
Mm-hmm.
Okay? So we know that endocrine disruptors are going to affect energy intake by affecting our hypothalamus. They know that some of them affect the levels of expression of MC4 receptors, for instance, in the hypothalamus. We know that they may affect absorption in the intestine. They affect our microbiota that also, can be involved in controlling our food intake and
Yeah.
satiety. And at the same time, we know that other chemical DDTs, one good example, can affect energy output by affecting thermogenesis in the brown adipose tissue. We know that many of these chemicals affect thyroid hormone action, and thyroid hormone has, of course, a good relation, an important relation with metabolism.
Yeah.
And what has been most studied is the effect in energy storage, how some of these chemicals affect the adipocytes, and they affect adipogenesis, and affect lipogenesis as well, uh, through nuclear receptors and other mechanisms, and promote energy storage. And at the same time, as we have been talking about, these chemicals change insulin levels, they change leptin levels, they change adiponectin levels, interleukin levels as well. And, it's a whole cocktail of effects disrupting every term and every tissue involved in energy balance equation. So predicting what the output is going to be, what the final phenotype is going to be, is very, very difficult.
Yeah.
But
Yeah.
I'm sure they affect, and your final phenotype is going to be somehow affected by all the chemicals you are exposed.
Right.
And mostly it seems to promote obesity, diabetes, and fatty liver as well.
Yeah. And, on one level, it makes perfect sense, right? Like there's, your propensity for obesity has many underlying terms that are going to matter, right? There's certain genetic
Mhm.
predispositions. We're all going to be genetically different to some extent.
Of course.
The energy balance equation, as you said, has multiple components. There's the food intake. There's the energy storage. There are things like energy expenditure and your basal metabolic
Mhm.
rate, and all of these variables can be affected to some extent or another by on any number of endocrine disruptors. And so,
Mhm.
if we're all exposed to a cocktail of these things, and it's a different cocktail for you and me,
Mhm.
and maybe I was exposed in the womb, but you were only exposed in adulthood, et cetera, et cetera,
Yeah.
all of those things are going to matter. And of course, there's not just one way to break metabolism and create an obese phenotype. So there's
Yeah.
not going to be one singular answer or one singular mechanism for what's causing obesity.
Yeah. That's part of the complexity of this field. That really we don't have a common mechanism of action for all endocrine disruptors.
Mm-hmm.
Uh, there are now a list of about 1,000 chemicals that are endocrine disruptors, or they may be endocrine disruptors. And we have more than 100,000 chemicals in commerce at the moment. So we don't know many of them, what the effect is. And all these chemicals are going to act very likely through different nuclear receptors, also other receptors at the membrane, or other pathways that we don't know.
Mm-hmm.
So that's really a very complex situation. And but this is how it is. This is life, and so this is why we are exposed to a cocktail and affecting perhaps not too much, but many different pathways, or practically all the pathways that are involved in our metabolism.
Yeah.
So it is difficult to predict, but it's very likely that this is changing the final phenotype. And see, for me, one of the main issues, of course, I work with insulin, but is changing the signals from insulin, leptin, and adiponectin, and all these hormones. that tell you the tissues how much you have been eating.
Hmm.
So, for instance, what we see with bisphenol A is that you have an over secretion of insulin. So that means you are, it's like telling your tissues that you are eating more than really you are doing, you know?
Mm.
If you eat something with bisphenol A, you're going to have more insulin release.
Yeah. Your body will
So
think you
that,
ate more than you
yeah,
actually did.
exactly. You are telling the adipocytes, okay, store more.
Yeah. Even though you don't need
Uh,
to store so
e-
much,
Even you don't
your
need to
cells
store so
think you need
much.
to store all that fat.
Exactly. The same to the liver.
Yeah.
And that hyperinsulinemia promotes insulin resistance, but also fatty liver and also obesity. So, changing the, the exact levels of those cues, hormonal cues, is really like, uh, I now imagine an example. It's like changing the time in traffic lights in the
Hmm.
middle of a big city like New York or Madrid, or
Yeah. Yeah.
even for a few milliseconds, it would be a disaster, at the very end.
When we think about, so I mean, these microplastics, these endocrine disruptors, they're in everything. We've created a world which is
Hmm.
filled with these things, and to a large extent, it's unavoidable that we're going to have some level of exposure to these things. What are some of the practical ways people can avoid exposure to things like microplastics and other endocrine disruptors? And in my
Hmm.
mind, there's sort of two categories of things I can think of. One, there's things you can do to prevent yourself from being exposed, and we can talk about some of those. But I also want to ask: is there anything that you can do to, anything you can eat, anything you can ingest that can prevent the microplastics from acting, soaks them up, and gets them out of your body, or something like that?
As far as I know, there is nothing. Of course, I mean, we can talk about individual things that we can do to avoid that. But I mean, there are some studies that are beginning to see whether some effects uh, of the plastic chemicals can be counteracted by other uh, phytoestrogens or other, part of diet that you can ingest and just counteract the effect in
Mm-hmm.
different type of cells. But as far as I know, it's nothing that we can really, uh, advise people to eat or drink that for you to get drink of to get rid of those microplastic or the chemicals in them. The thing we can do is try to avoid eating them as far as we can. So try to avoid, if you can, uh, food in plastic,
Mm-hmm.
uh, if it's possible, because it's not easy. I know. Try to buy organic food as well if you want to get rid of pesticides that we didn't talk about, but they are, of course, also endocrine disruptors as well. Uh, Try to open the windows in your house. This is very easy, and just have fresh air during 20 minutes, as we did during the COVID pandemic, just to get rid of the volatile endocrine disruptors that are also in furniture and in plastics at home.
So
I mean, that's
there's
yeah,
volatile
yeah,
compounds. Wow.
yeah, yeah, absolutely. Yeah, they're everywhere. Small concentrations, but they do a small effect. That
Mm-hmm.
if we add, then we may have a problem, as we said. So we can do these individual things, but I want to point that really this is a global problem, and it needs a global solution. This is something that goes in parallel with climate change, in the sense that it's all part of the petrochemical, you know, uh, use for energy and for many other components. And the same way that climate change is approached by international entities, the chemical pollution, including the endocrine disruptor pollution, has to be also approached uh, as a global trying to give a global solution as well to this problem.
What are what are some of the things you guys are working on in the lab today? What are some of the questions related to endocrine disruptors that you're investigating right now that we'll probably start to get answers to in the next couple of years?
Yeah, at the moment, yeah, we are focused in two different projects. One is related to what we talk about, why bisphenols have a high efficacy with a low affinity for the
Mm.
receptors.
So even though they don't stick to the receptor much, they are
Yeah,
able to turn on the receptor essentially quite a bit.
yeah, they turn the receptor on enough time to produce a signal. And this is because there is a proton transfer from the ligand to the ligand binding domain. And
Mm-hmm.
we think this is what is switching this extranuclear action. So we're working on that. And we have, as I said, a paper that we are in the last stages of we hope to publish in the next month. It's already a preprint published uh, with this, and we are trying to study that with different ligands and with other and with nuclear receptors other than estrogen receptors. So that's one of the projects. And the other project, which is together with Paloma Alonso Magdalena, who is uh, we are colleagues collaborating for many years, is about pregnancy. What are the epigenetic mechanisms that happens in the mother
Mm-hmm.
later in life in order to have this phenotype of altered glucose homeostasis? So we know that this phenotype is happening, but we like to understand what is the mechanism or are the mechanism driving to those changes in the pancreas, the adipose tissue, and
Hmm.
the liver.
Hmm.
So these are the two projects we are involved now.
Interesting. How has working in this field changed your life on a personal level? Like, are you I don't know, do you use the same shampoo? Do you use the same body lotion? Has it completely changed all of the products that you're using?
No, I haven't changed all of the products, but of course, I mean, I don't exaggerate about this too much on myself, but I take care. I think it is important. So uh, I I never I try to eat nothing that is in touch with plastic.
Hmm.
So I prefer to go to the grocery shop and buy ham or cheese or whatever, and they cut it and give it to me,
Hmm.
and not with plastic wrap, you know.
Yeah, yeah.
I mean, these sort of things. I don't drink from plastic bottles, never. So I use uh, normally a bottle, metal bottle, and and tap water to drink. Filter tap water. It's, and with the shampoo and try to avoid parabens and as much as I can read, but this is not easy.
Yeah,
That I
yeah.
try to do is to, for instance, I use solid shampoo to avoid the plastic bottle.
Ah, I see.
And for shaving the same. I don't use the plastic shaving machines. So I normally use a metal blade, and it's
Hmm.
like the old fashioned,
Yeah, yeah.
which is not so nice, to your skin, but it works. And I avoid, plastic as much as I can.
Yeah.
So that's all. But I want to emphasize one personal change that I did years ago, uh, because I think the important period to avoid exposure is pregnancy and infancy.
Mm.
Because during pregnancy is when the fetus is going to, have more changes that may last for the whole life.
Yeah.
So when I had my first child, I didn't know about BPA. or the effects they did. That was in 96. So we use plastic bottles,
Mm.
polycarbonate plastic bottles.
Hmm.
Were there? Okay. My second child, we already did the experiments with BPA. He was born in 99. So we avoid all plastic bottles. Later, plastic bottles were banned in Canada, here in Europe, in Japan, and many other countries uh, since 2010, 2014, more or less,
Hmm.
you know. But that was a personal, I think, big change that I recommend
Yeah.
for
Just
everyone,
being
just in
extra
case,
vigilant during those critical periods.
try to avoid exposure as much as you can during pregnancy and infancy,
Hmm.
because it's when most of the epigenetic changes are going to happen, and we know that already.
Yeah. Well, we've covered a lot already. Again, this is this is a fascinating and terrifying area of study, but obviously very important and relevant to everyone. Are there any final thoughts you want to leave people with, or any things you want to reiterate from our discussion?
Yeah, I want to just to say that, as I said, this is a global problem, and I think at least here in Europe, the European Commission is aware of it. In the Green Deal for 2030, the European uh, Union, the European Commission said that we should get rid of toxic chemicals and we should have a zero toxic environment, including endocrine disruptors. I don't know if this is going to happen by 2030. Now the European Union members of the parliament have changed, and we don't know what is going to happen in the next 4 years. I hope they continue with this willing in order to fight from, you know, in order for our politicians to fight this problem and try to give a global solution here in Europe.
Hmm.
Mm, and I hope this is somehow translate to other countries as the States, that I know you have different regulation, perhaps not so strict as we have in Europe. And I want to give this message in the sense, okay, we know that politicians are aware. We scientists work with scientific societies, for instance, the Endocrine Society and the European Society of Endocrinology are working very much with policymakers,
Hmm.
trying to uh, decrease at least this problem, decrease exposure to endocrine disruptors, and to change policy making to avoid what we just been talking about.
Yeah.
And yeah, I want just to give this message that it's going very slowly, but I hope it continues. And one day, I don't know if I will see it, but we had really zero toxic chemical environment.
Hmm. All right. Well, Professor Angel Nadal, thank you very much for your time. This was fascinating.
Thank you very much, Nick. Had been a great pleasure talking to you.